Hello, welcome to pH Biology. Today's explainer is denaturation, the change in shape of an enzyme caused by heat or extreme pH. By the end of this video, you'll know what causes it, why it's usually permanent, and how it shows up on every enzyme rate graph. Denaturation is a change in the shape of an enzyme, particularly its active site, caused by heat or extreme pH. The bonds that hold the protein in its three-dimensional shape, especially hydrogen bonds and ionic interactions, break. The active site no longer fits the substrate and the enzyme stops catalyzing the reaction. Three things. 1. Denaturation is usually permanent. Cooling a denatured enzyme back to body temperature doesn't restore the active site shape. 2. Different enzymes denature at different temperatures and pH values. 3. On a rate temperature graph, denaturation appears as the sharp drop in rate above the optimum. Common slip. Say denatured, not died or killed. Enzymes aren't alive, and don't say the enzyme is broken. The amino acids are still in the right sequence, but the three-dimensional shape has changed. Without that shape, the enzyme is useless. Quick example, frying an egg. The clear gel of egg white is mostly the protein albumin in solution. Heat the egg and the albumin denatures. The protein unfolds, tangles with neighbors, and the clear liquid becomes opaque and solid. Same molecular process as denaturing a digestive enzyme. Right, if any of this is going to stick beyond this video, drop into phmaths.com. You'll find a free 4-week GCSE higher revision planner ready to download, plus the mastery workbooks. One for every higher topic, every answer fully worked, every common slip flagged. Simple explanations, serious results. See you next video.